Glycolysis, also known as the Embden-Meyerhof pathway, is a pathway for carbohydrate catabolism that begins with the substrate D-glucose. The pathway evolved at a time when the Earth's atmosphere was anaerobic. No free oxygen was available. As a result, glycolysis requires no oxygen. It is an anaerobic process. Further, it must have evolved in very simple, single-celled organisms that lacked complex organelles, much like bacteria. As a result, glycolysis is a process carried out by enzymes that are free in the cytoplasm. To this day, glycolysis remains an anaerobic process carried out by cytoplasmic enzymes, even in cells as complex as our own. The 10 steps of glycolysis are catalyzed by 10 enzymes. The first reactions of glycolysis involve an energy investment. ATP molecules are hydrolyzed, energy is released, and phosphoryl groups are added to hexose sugars. In the remaining steps of glycolysis, energy is harvested to produce a net gain of ATP. The three major products of glycolysis are chemical energy in the form of ATP, chemical energy in the form of NADH, and two 3-carbon pyruvate molecules. 4 ATP molecules are formed by the process of substrate-level phosphorylation. In this process, a high-energy phosphoryl group from one of the substrates is transferred to ADP to form ATP. The two substrates involved in these transfer reactions are 1,3-bisphosphoglycerate and Phosphoenolpyruvate. Although 4 ATP molecules are produced during glycolysis, the net gain is only 2 ATP molecules because 2 ATP molecules are used early in glycolysis. Nicotinamide adenine dinucleotide, or NAD+, is a coenzyme derived from the vitamin niacin. The reduced form of NAD+, NADH, carries hydride anions, hydrogen atoms with 2 electrons, removed during the oxidation of 3-glyceraldehyde 3-phosphate. Under aerobic conditions, the hydride is transported from the cytoplasm into the mitochondria. Here it enters an electron transport system for the generation of ATP by oxidative phosphorylation. Under anaerobic conditions, NADH is used as a source of electrons in fermentation reactions. At the end of glycolysis, the 6-carbon glucose molecule has been converted into two 3-carbon pyruvate molecules. The fate of pyruvate also depends on whether the reactions are occurring in the presence or absence of oxygen. Under aerobic conditions, it is used to produce acetyl coenzyme A, destined for the citric acid cycle and complete oxidation. Under anaerobic conditions, it is used as an electron acceptor in fermentation reactions. Both pyruvate and NADH must be used in some way so that glycolysis can continue to function and produce ATP. The buildup of pyruvate would cause glycolysis to stop, thereby stopping the production of ATP. If NADH were to build up, there would be no NAD+ available for step 6, in which glyceraldehyde 3-phosphate is oxidized and NAD+ is reduced. This would stop glycolysis, and thus NADH must be reoxidized so that glycolysis can continue to produce ATP for the cell. In this figure, you can see the products of the glycolysis reactions. We form 4 ATP molecules, but we use 2 ATP molecules in the first 3 steps, therefore the net gain of ATP is only 2 ATP. We also produce NADH and pyruvate. Glycolysis can be divided into 2 major segments. The first set of steps, reactions 1 to 5, is the investment of ATP energy. The second major segment involves the remaining reactions of the pathway, steps 6 through 10, those that result in a net energy yield. In reaction 1 of glycolysis, the substrate glucose is phosphorylated by the enzyme hexokinase in a coupled phosphorylation reaction. The source of the phosphoryl group is ATP. At first, this reaction seems contrary to the overall purpose of catabolism, the production of ATP. The expenditure of ATP in these early reactions must be thought of as an investment. The cell actually goes into energy debt in these early reactions, but this is absolutely necessary to get the pathway started. Here's my 6-carbon sugar, glucose, and my adenosine triphosphate. The enzyme is hexokinase, and kinases catalyze phosphorylations. And what we're doing is we're adding a phosphoryl group on the 6th carbon. The products are glucose 6-phosphate, G6P, and ADP. The glucose 6-phosphate, or G6P, formed in the first reaction is rearranged to produce the structural isomer fructose 6-phosphate. The enzyme phosphoglucose isomerase catalyzes this isomerization. The result is that the carbon-1 carbon of the 6-carbon sugar is exposed. It is no longer part of the ring structure. Examination of the open chain structures reveals that this isomerization converts an aldose into a ketose. So carbon 1 in the G6P molecule is a secondary alcohol. Here's the hydroxyl. But when it isomerizes to give you F6P, or fructose 6-phosphate, you can see that this carbon is this one here. And now we have a primary alcohol, which is going to be more exposed. The enzyme that catalyzes the reaction, phosphoglucose isomerase, simply isomerizes the molecule. If you look at the molecular formula of G6P and that of fructose 6-phosphate, you'll see that they're identical. All that's happening is isomerization, or rearrangement of the structure. A second energy investment is catalyzed by the enzyme phosphofructokinase. A phosphoanhydride bond in ATP is hydrolyzed, and a phosphoester linkage between the phosphoryl group and the carbon-1 hydroxyl group of F6P, or fructose 6-phosphate, is formed. The product is fructose 1,6-bisphosphate. In reaction 3, another kinase transfers a phosphoryl group from ATP onto the hydroxyl at carbon 1 to give us fructose 1,6-bisphosphate. The abbreviation that's used for phosphofructokinase is PFK. Fructose 1,6-bisphosphate is then split into two 3-carbon intermediates in a reaction catalyzed by an enzyme aldolase. The products are glyceraldehyde 3-phosphate, or G3P, and dihydroxyacetone phosphate, or DHAP. So fructose 1,6-bisphosphate is a 6-carbon molecule, and aldolase serves to split it in two. We end up with dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or G3P. Because G3P is the only substrate that can be used by the next enzyme in the pathway, the dihydroxyacetone phosphate is rearranged to become a second molecule of G3P. The enzyme that mediates this isomerization is triose phosphate isomerase. Again, the substrate in reaction 6 you're going to see is only G3P. So that means that we have to convert any DHAP into G3P. This is done via an isomerization, and the enzyme that catalyzes the reaction is triose phosphate isomerase. Just like the isomerization in reaction 2, if you look at the molecular formula of DHAP and you compare it to that of G3P, you'll see that they're identical. All that's happening is a rearrangement of the molecule. In reaction 6, the aldehyde glyceraldehyde 3-phosphate, or G3P, is oxidized to a carboxylic acid in a reaction catalyzed by. In reaction 6, the aldehyde G3P, or glyceraldehyde 3-phosphate, is oxidized to a carboxylic acid in a reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. This is the first step in glycolysis that harvests energy, and it involves the reduction of the coenzyme nicotinamide adenine dinucleotide, or NAD+. This reaction occurs in two steps. First, NAD+ is reduced to NADH as the aldehyde group of G3P, or glyceraldehyde 3-phosphate, is oxidized to a carboxyl group. Second, an inorganic phosphate group is transferred to the carboxyl group to give 1,3-bisphosphoglycerate. Notice that the new bond is denoted with a squiggle, indicating that it is a high-energy bond. Not only does G3P get oxidized from the aldehyde to a carboxylic acid, but it also undergoes a phosphorylation to give us 1,3-bisphosphoglycerate. We also harvest energy in the form of NADH. Reaction 6 and all remaining reactions of glycolysis occur twice for each glucose because each glucose has been converted into two molecules of glyceraldehyde 3-phosphate. In reaction 7, energy is harvested in the form of ATP. The enzyme phosphoglycerate kinase catalyzes the transfer of the phosphoryl group of 1,3-bisphosphoglycerate to ADP. This is the first substrate-level phosphorylation of glycolysis, and it produces ATP and 3-phosphoglycerate. It is a coupled reaction in which the high-energy bond is hydrolyzed and the energy released is used to drive the synthesis of ATP. Here we see the high-energy phosphoanhydride bond, which is used to phosphorylate or add a phosphoryl group to adenosine diphosphate and produce ATP. In reaction 8, 3-phosphoglycerate is isomerized to produce 2-phosphoglycerate in a reaction catalyzed by the enzyme phosphoglycerate mutase. The phosphoryl group attached to the third carbon of 3-phosphoglycerate is transferred to the second carbon. The phosphoglycerate mutase enzyme is simply responsible for the isomerization of 3-phosphoglycerate to 2-phosphoglycerate. So really all we're doing is taking this phosphoryl group and moving it from the oxygen on this carbon to this carbon. If you look at the molecular formulas of these two compounds, they are identical. In reaction 9, the enzyme enolase catalyzes the dehydration or removal of water of 2-phosphoglycerate. The energy-rich product is Phosphoenolpyruvate, the highest energy phosphorylated compound in our metabolism. In 2-phosphoglycerate, the atoms that comprise water are highlighted, and when we lose water, we end up forming a pi bond to make phosphoenolpyruvate. Reaction 10. The final substrate-level phosphorylation in the pathway is catalyzed by pyruvate kinase. Phosphoenolpyruvate serves as a donor of the phosphoryl group that is transferred to ADP to produce ATP. This is another coupled reaction in which hydrolysis of the phosphoester bond in phosphoenolpyruvate provides energy for the formation of the phosphoanhydride bond of ATP. The final product of glycolysis is pyruvate. Reactions 6 through 10 occur twice per glucose molecule because the starting 6-carbon sugar is split into two 3-carbon molecules. Thus, glycolysis produces 2 NADH molecules and a total of 4 ATP molecules. The net ATP gain from this pathway is, however, only 2 ATP molecules because of the energy investment of 2 ATP molecules in the early steps of the pathway. Depending on the tissue, fructose enters glycolysis in different ways. In the muscle, where hexokinase is abundant, the enzyme phosphorylates fructose to fructose 6-phosphate, which directly enters glycolysis. There is much less hexokinase in the liver, but fructokinase is present. Fructokinase phosphorylates fructose to produce fructose 1-phosphate. This product is cleaved into dihydroxyacetone phosphate, or DHAP, and glyceraldehyde by the enzyme fructose 1-phosphate aldolase. The glyceraldehyde is phosphorylated by triose kinase to produce glyceraldehyde 3-phosphate, G3P. The DHAP and G3P enter glycolysis directly.